Okay, so I got a brief presentation here, just because I want to, kind of help tie a few subjects together that we've been talking about, and specifically talk about parthenogenesis and meiosis. And we talked about, for example, a lot of examples of snakes that produce babies parthenogenetically, and so I wanted to kind of walk through the steps of meiosis and show you how this can happen, because I think it's interesting and it helps to, again, this is why we want you to understand the steps of meiosis and mitosis, because then you can sort of explain and understand things that you see in nature. And so parthenogenesis is rare, but it's not unheard of in vertebrates, right? And again, the examples in the Louisville Zoo is that Thelma the python, who produced a bunch of eggs, and they hatched, and they were produced without a male. They were unfertilized eggs, yet they produced viable offspring. Now, sometimes when this happens, the parthenogenetic clone, parthenogenetic offspring is known as a half clone of the mother. And so what does this mean, and how does this happen? And so I kind of made this and kind of worked through meiosis. So this is how I think it happens. I might be wrong, but I think I'm right. So let's go back to meiosis, and here's just a simple example of an organism whose diploid number is 6. So it's got 3 homologous pairs of chromosomes. And on the left, you've got G1 of interphase. And then on the right, you've got that same cell in G2 of interphase. So after S phase, so where each of those chromosomes has been replicated, but you still have the diploid number of chromosomes, right? 6 chromosomes. And in this example I'm going to show you, the maternal chromosomes are red and the paternal chromosomes are blue. And so for each homologous pair, you've got a red one and a blue one, right? You've got 1 chromosome from mom and 1 chromosome from dad. Also, again, I want to note that I realize this is interphase, and typically during interphase you can't see the chromosomes, but this is just an example, right? So we want to kind of pay attention to these red and blue chromosomes and kind of help us understand how we can form what's known as a half clone through parthenogenesis. Okay, so now we've got that same cell and we're saying we're in prophase 1. So again, we can see the chromosomes, they've condensed. We got the diploid number of chromosomes. And then those go into metaphase 1, where the homologous chromosomes line up. So this is meiosis. And so the first thing we're going to do is separate those homologous chromosomes. And they're lining up in the middle. Chromosomes go to the middle during metaphase. Now notice how they pair up and how they line up at the equator. Sometimes the blue chromosome is on the left, and sometimes the blue chromosome is on the right. And it's random how they line up. And so sometimes the paternal chromosome is on one side, and sometimes the paternal chromosome is on the other side. And of course the maternal chromosome is opposite of that. Remember that we call this independent assortment. How one homologous pair lines up at the equator is independent of how all the other ones line up. And so when Mendel came up with this idea of independent assortment, he saw that some traits are passed on independent of other traits, and this is why. Because those traits are on different chromosomes. Now, sometimes you see traits that are linked, and so the traits tend to be inherited together. Well, a lot of times that's because they're on the same chromosome. But it's this idea that homologous pairs line up independent of one another, and that's the paternal and maternal chromosomes are independent of other paternal and maternal chromosomes. Okay, so again, we're repeating, we've got metaphase 1 on the left here, and then during anaphase, they get separated. Anaphase 1 is where we separate these homologous chromosomes. So notice how on the left, in anaphase 1, you've got a paternal chromosome and 2 maternal chromosomes. Whereas on the right, in this anaphase 1 cell, you've got 1 maternal chromosome and 2 paternal chromosomes. You see that? We're just separating the blues and the reds here. And we're talking about, pulling them apart in the anaphase. Now, after anaphase, we have telophase and then telophase and then cytokinesis. And so then we end up with 2 cells. But we're talking about parthenogenesis here. So we're talking about unfertilized eggs. So we're making eggs. And you'll remember that a lot of times when we make an egg, we don't divide the cytoplasm equally. That one cell will get most of the cytoplasm, and the other, just has DNA and a little bit of cytoplasm. And that small one we call a polar body. And the polar bodies usually don't develop any further. They just get reabsorbed. And of course what we're doing is we're parceling all the cytoplasm. We're going to end up with most of the cytoplasm in one egg. So instead of producing 4 haploid eggs, we're only going to produce 1 big haploid egg. So here we made our first polar body, and it may go through meiosis too, but ultimately this is just going to be reabsorbed. It's not going to form a viable egg. Okay, so now we've got that 1 egg in metaphase 2, which is shown here, and now we're going to rip apart those sister chromatids in anaphase 2. And so that's what we're showing here, is how those sister chromatids are ripped apart. And so technically, during anaphase 2, this cell is diploid, right, for a split second. So it was haploid. In metaphase 2, again, count centromeres. You have 1, 2, 3 centromeres. You've got 3 chromosomes. It's just that each chromosome has been duplicated. In anaphase 2, we've ripped apart those sister chromatids, and so now count centromeres. 1, 2, 3, 4, 5, 6. We actually have 6 chromosomes now during anaphase 2. Again, normally, we would go to telophase 2 and cytokinesis 2, and we'd end up with 2 cells, but again, 1 cell gets all the cytoplasm, the other one gets nothing. It just has DNA and a little cytoplasm. And so then that's again a polar body, which is just going to be reabsorbed, and we're left with 1 big haploid egg, and this is typically how it happens. But you'll remember that a lot of eggs will pause during meiosis 2. And so they'll pause at this point where they've got, they're technically diploid. Now normally then that egg gets fertilized, and once it gets fertilized, that stimulates it to wrap up meiosis 2 and kick out that polar body, and you're left with, the male's DNA, the haploid number of the female's DNA, and so your fertilized, your zygote has the right amount of DNA. But what if there was no cytokinesis and no telophase 2 after anaphase 2? And so what if that cell stopped in meiosis 2, no sperm came in, but no cytokinesis happened either? So the polar body does not get ejected. The polar body does not get reabsorbed. Okay? Technically, at this point, you have a diploid organism. And so if this cell, which is diploid, starts going through mitosis, well, then it can form a regular diploid offspring. And that's how you can get parthenogenesis. That's how you can get an egg that can develop into a diploid offspring without bringing in any DNA from a sperm. And so that's a pretty typical way that this happens. And in these snakes, this is probably what happens. Instead of having cytokinesis, you just start going through mitosis, and now you've got a diploid organism. So now look at the color of the chromosomes and think about their relationship, right? You've got these red chromosomes and blue chromosomes that came from the paternal and maternal. Red is maternal, blue is paternal. Now, if this cell starts going through mitosis and dividing, that red is actually from, it's its grandmother's chromosome, and the blue is its grandfather's chromosome, is another way to think about it. But also, again, think where these particular chromosomes in this example came from. This is anaphase 2, where we separated sister chromatids. Those sister chromatids were identical. Now, if those sister chromatids become the new homologous pair, that means each homologous pair is made of identical chromosomes. That's not how it typically works, right? All these homologous pairs were sister chromatids at one time, so they are exactly the same. So if this is a parthenogenic organism, it's going to have the same chromosome at every homologous pair. And so let's compare that to a regular organism. Let's compare that to mom. So on the right here, we've got mom's original cell during prophase 1, and I've marked the homologous pairs. And so you see in each homologous pair, you've got a red chromosome and a blue chromosome, which is typically what happens, right? You get one from your mother and one from your father, and that's what the mom in this example did. But in the parthenogenic cell, you either, for each homologous pair, you either get both the maternal chromosome or both are the paternal chromosome. You don't have this mix of maternal and paternal like you do in a typical organism. And so that makes the parthenogenetic offspring unique from other offspring. And so you can look at, you have unique inheritance patterns because of the way the chromosomes are parceled out. And so then why do we say that this is a half clone of the mother? I mean, don't all offspring, have half their mother's DNA? So how come a regular offspring that gets half its mother's DNA, we don't call that a half clone? Well, I think, in a regular offspring, half of those chromosomes all come from the father. And so, yes, half the chromosomes come from the mother, but half come from the father, and at each homologous pair, it's one from the mother and one from the father. But in these parthenogenetic offspring, it's for every homologous pair, they're either both are maternal or both are paternal. And again, we're talking about for that offspring, it's actually its grandmother or its grandfather's chromosome. But the point here is, in a typical offspring, half the DNA is from mom and half of the DNA is from dad, but in these parthenogenetic offspring, they all came from the mother. And so that's where I think we get the clone part. It's a clone because there's no father involved. It's all DNA from the mother. But we call it a half clone because it's not identical to the mother. Because of the way the meiosis worked, and because of the way the parthenogenesis worked, at every homologous chromosome, it's either the maternal chromosome or the paternal chromosome, but it's not a mix of both like it is in the mother. You see what I'm saying? So I think the clone part is because all the DNA comes from mom, but it's a half clone because it's not identical to the mother. And so again, here on the right, we have mom's cell. And for each homologous pair, it's one red and one blue chromosome. And so if it was a full clone, then the offspring would have the exact same chromosome. The offspring would have one red and one blue chromosome. But in the parthenogenetic cell, we don't have that. For every homologous pair, it's either red or blue. They're both red or blue. Okay? So study how these move and what those colors mean, and I think you'll understand what I'm talking about. Now again, if you study this, you can see why the parthenogenetic cell will have high levels of homozygosity. So what is homozygosity? Homozygous is when you have two alleles that are the same. In this parthenogenetic offspring, their homologous pairs were formed by splitting sister chromatids. Sister chromatids are identical. So if you have an allele on one chromatid, you've got the same allele on the other chromatid. And so then if these end up making your homologous pairs, as they do in this example, well then you're going to have the same alleles on every chromosome. So you're going to be homozygous at all those gene loci. And so that's why parthenogenetic offspring have high levels of homozygosity. So, once you kind of understand the steps, then all these other cool things all start to make a lot more sense. Okay, and so in conclusion, now what's something that occurs during meiosis that we haven't talked about? Remember, crossing over during prophase 1 is a big deal. Okay, that's going to muddy those results some, and that's going to affect homozygosity. I'm going to leave that to you to think about. I'm not going to tell you. But think about, how will crossing over influence this idea of high homozygosity in the parthenogenetic offspring? Think about that. It's interesting. Now, I think I'm right about the definition of half clone and full clone. Maybe I'm wrong, but I think I'm right. But the definition of half clone or full clone is not as important as understanding how those chromosomes move and understanding how, although this parthenogenetic offspring sprang from the mother and it has only DNA from the mother, so there's no other male or no other individual contributing to this offspring's DNA, so all its DNA came from mom, yet it's not a full clone of its mother. Well, you can understand that if you follow those chromosomes and you can see how the homologous pairs are not identical. Even though they all came from mom, those offspring are not full clones of their mom. And so that's what's important, is understanding how those chromosomes move. And again, this just points out, this is why we want you to know all these things, because by understanding the steps, like to things like meiosis, now you start to understand, the really interesting stuff that you find out there in biology. So anyway, I thought this was really cool. I hope you did too. I hope I didn't confuse you too much, but I tried to do this step by step. So go through this a couple times and you can see how those chromosomes move, and I think it'll make sense to you. But anyway, if you've got questions, as always, let me know. That's all I've got for here. So I'll talk to you later. See ya.